Hello everyone, we are solving question number 4 of GATE 2011 and it says yeast artificial chromosome that are YACs are used for cloning large segment of DNA, mRNA, bacterial DNA or yeast DNA. So let's see uh, how YAC and BACs work. So for YAC, their host cell is yeast, that is Saccharomyces cerevisiae, but, but for BACs, the host cell is E. coli. So YAC, the type of DNA molecule is linear chromosome, and the insert size is up to 1 to 2 uh, megabases. But in case of BAC, you have circular plasmid and it can take up to 300 kilobases. So the protocol for handling and isolation of DNA is little difficult in case of YAC, but it is easy in case of BAC. The DNA yield is low and resistance to shear is very low. Then direct sequencing is not possible. There are some selection marker that are specific to yeast growth, and but in case of BAC, you have DNA yield high or medium, as it is easy to handle, and the protocol is little easy as compared to YAC. Direct sequencing is possible in case of BAC. So the answer for the question is it is used for the large segment of DNA. But just to give another aspect for all cloning vectors. If we talk about the vectors that we use, there are two types of vector. One is cloning vector and expression vector. So cloning vectors are the vectors we used for cloning the required gene of interest or DNA segment, and expression vector is used for the expression of a particular gene in in terms of protein. So cloning vectors, for smaller size, we use PBR32 and PUC. These are E. coli based vector. Then we have cosmid vector, which is PGB8. Then phagmide vector, bacteriophages. They are also uh, vectors that can clone a small segment of DNA. But when it comes to large segment of DNA, we use PAC, BAC, and YAC. YAC can carry the larger segment as compared to BAC and PAC. So this is for different type of vector.